Welcome to the Beer Taster's Notebook, the show focused on beer education. I'm Jonathan Thompson, Advanced Cicerone, BJCP beer judge, and fermentation enthusiast. Okay, welcome back to the Beer Taster's Notebook, episode 19, Hops and Bitterness. So we're going to jump into hop bitterness and hit it from three different angles. First of all, what is bitterness? Secondly, why would a plant want to produce such a thing? And thirdly, how does it get into our beer? So, bitterness is one of the 5 established and accepted tastes. We talked about all of this in the taste episode, but we can rehash it here a little bit. So, I guess defining bitterness is kind of like defining thoughts. Maybe not quite that vague, but generally we agree that it's a taste. In other words, it's a chemical reaction that sends a signal to the brain. And so humans need various compounds to survive. So we need glucose for energy. We need proteins, or I guess more specifically amino acids for our bodies to build stuff. And we need fats as well for energy, for various functions, including building cell walls, without which we'd have some major issues. We also need salt in order to maintain fluid balance. But what we don't historically need are poisonous plants and spoiled food. And so this leads us to why one might naturally be attracted to cheesecake, or I guess unnaturally really, because they don't exist in nature, but you get what I'm saying here, versus like a Brussels sprout or sour kefir. And we've, of course, programmed ourselves now to enjoy bitter compounds like chocolate or coffee, although to be fair, not so many people eat the pure unsweetened chocolate, which is my favorite. And a high number of people, just to put it mildly, will add sugar and or milk or cream to their coffee. As a side note, I actually think adding cream to coffee has some serious culinary and scientific merit, but we can explore that maybe in 2045 when I run out of beer stuff to talk about. So acidity can also be pleasant, obviously, when one is used to it and when it's used sparingly. But of course, you're more likely to be presented with like a sugar-laden lemon loaf, let's say, than just a hunk of raw lemon, when you join your friends for high tea. Okay, so just back to sort of the biology a little bit. So bitterness is a defense system. Evolution has gifted us and many a creature with the receptors to detect it. And speaking for the humans, the bitter compounds will bind to a G protein coupled receptor called TAS2R. And after such binding, will undergo a conformational change, sending a signal to the brain. It's usually a Danger Will Robinson sort of signal. And most importantly, these receptors reside on the tongue, of course, in the taste buds, but in other places in the mouth as well, and throughout the body, including some very strange areas. And so that's the basic pathway of mouth to brain, and then back to the mouth, in the case of us sort of spitting it out or rejecting the food. And as this might be the bane of many a parent, it's kept us alive as a species for a long time. And so Brussels sprouts are good for us, right? Watercress, which can be bitter in the stems, really is one of the most nutritionally dense foods out there. Kale and Swiss chard and broccoli obviously have a lot of nutritional merit. So I guess the question is, why do our bodies want us to reject these? And they don't specifically, but they also do. And so going back a little bit in time and just talking about plants, the purpose of any species, and I say this a lot, is the continuation of the species. So in other words, the passing on of genetic material. This really boils down to sex and survival, but physical health is paramount to survival as well. And so when selecting a mate, one tends to look for a viable partner and looks for traits that we might desire to be passed on to the next generation. Humans, especially now, are highly suggestible, and so this doesn't always track really, as they would say, right? So but underneath a fairly manipulated psyche, that drive is still there. And so back to plants, they don't have Instagram and perfume ads, and so they remain relatively intact as far as their selection goes. But for now, we just need to focus on bitterness. So in order to stay alive, considering too that they can't really run away, they need to develop some defenses. As well as some way to obviously continue on. So fruits with their bright colors and alluring smells attract animals who eat them and either dispose of the seeds directly or later on through defecation, spreading this material around and obviously propagating the species, et cetera, et cetera. So for the parts that need to remain intact, though, they need defenses. And so thorns, trichomes, like the hairs on a singing nettle, cactus needles, the bad smells, et cetera, are all ways of keeping predators at bay. Toxins, of course, are another. And so at one point, plants noticed that bitter alkaloids, which were toxic and off-putting, of course, were keeping these other plants from being eaten. And so in a fit of jealousy, they decided they would figure out ways of developing their own. And so it's kind of the same thread as producing smells to trick or entice bees. So they started developing these bitter compounds as a deterrent, but bitter compounds that were not actually toxic. And so when an animal encounters a stimulus akin to death, they tend to back off. Humans were clearly stupid or smart enough, really, I guess, to test these waters a little more aggressively. Had we not, we'd have no shaved Brussels sprout salads, Negronis, half grapefruit and black coffee diet trends, and more importantly, we wouldn't have beer, or at least we wouldn't have beer as we know it. And so bitterness comes in a few forms in plants, mainly found in terpenoids, flavonoids, and some glycosides. And then the most commonly associated version in alkaloids. Caffeine, by the way, is an alkaloid, so are morphine and nicotine. And so let's move on to hops a little bit here. So we've established now that plants use these bitter compounds as a defense mechanism, and so hops are no different. They produce these bitter acids to keep things from eating them. Unfortunately for them, we have a lot of beer drinkers out there, and so the hop's fate is pretty much sealed. Anyways, so we call these bitter acids. They're the alpha and beta acids, so aka humulones and lupulones. And so let's talk about what they are specifically, how the plants make them, and how they get into the beer. So let's start off with the alpha acids. They are the most relevant. And so the most relevant of the most relevant are humulone, adhumulone, and cohumulone. Prehumulone and posthumulone are minor players, but they're nonetheless in existence. So humulone, cohumulone, and adhumulone are the most common analogs, as mentioned. They are chemically quite similar, with differences in their side chains, specifically the acyl group, where humulone has an isovaleryl side chain, cohumulone has an ad-isobutyryl side chain, and adhumulone 2-methylbutyryl group. And so pre and post-humulone, by the way, are very minor, as I mentioned, and the main difference between the two of them is the number of carbons in their side chains. And so of these three main acids, humulone and cohumulone, they really take the majority share, 20 to 50% each, leaving 10 to 15-ish for adhumulone and then scraps for the pre and post. And so I guess the structure can be a little bit daunting here and probably unnecessary knowledge. So I think let's go into some of the formation basics, and I'm going to try and keep this as rudimentary as possible because it can get quite nuts. But basically, the first step is to form an acyl-CoA precursor. These are built from amino acids, and the amino acid itself actually determines the end product. So leucine forms isovaleryl-CoA, and then humulone as an end product. Valine, isobutyryl-CoA, and then cohumulone, and isoleucine, 2-methylbutyryl-CoA, and then, of course, adhumulone. And so the formation is via the transamination to alpha-keto acids, the oxidative decarboxylation, and then the formation of acyl-CoA thioesters. The next step we need is to create the fluoroglucanol color. This is done via valerophenone synthase, and then there are a bunch of steps, but the product here is a fluorisovalerophenone or an analog of such things. And so the next step is the prenylation, and this is the addition of hydrophobic components to a protein or other biomolecule, whereby prenyl transferase acts upon the PIVP along with dimethylallyl pyrophosphate, DMAPP for short, forming mono and deprenylated intermediaries. And then through some oxidation and ring arrangements, we end up with the alpha acids. And so the formation of the lupulones or beta acids are much the same, although they have 3 prenyl groups versus the alpha acids 2. And they have different enzymes which act upon these precursors. So let's deal with the beta acids first and their contribution to beer, and then the alphas and isomerization. So beta acids make up only 3 to 10% of the acids. They provide a smoother bitterness, so to speak, but the pathway is a lot different. And so while alpha acids isomerize, as we'll discuss shortly, the beta acids don't, and they actually oxidize over time into hulupones, which are soluble. And this is important in aged beers because the alpha acids will degrade over time. And so as they're degrading, the beta acids are oxidizing and then sort of lending a hand to the bitterness in the bitterness chain. And so of equal importance is that the beta acids are responsible for most of the antimicrobial properties of hops. And so these are pretty effective against gram-positive bacteria. And let's just touch on that quickly, just so you know. So gram-positive and negative really have to do with staining. Well, they don't, but that's where the names come from. So it's the name Gram stain comes from Hans Christian Gram, who was a Danish bacteriologist. And so basically they use, first of all, a purple stain, and gram-positives will stain purple and remain stained purple, while gram-negatives will not when it's washed out with alcohol. And then a counter stain is applied, and the gram-negative will push out the purple stain and stain pink. And so gram-positive bacteria have a thick peptidoglycan cell wall, and it retains the stain, whereas gram-negative have a thinner one, which obviously allows it to be washed out. And so the beta acids will work well on gram-positives, and this is important defense against things like lactobacillus and pediococcus, which can contribute to beer spoilage pretty quickly. Interestingly, they also have a detrimental effect on Propionibacterium acnes, Staphylococcus aureus, which contribute to acne, and on Listeria monocytogenes. And so the hop acids have basically limited to no effect on gram-negative bacteria, as we mentioned, like Salmonella. So despite the presence of these, hygiene is obviously key in brewing. The way they work, by the way, is to disrupt the proton motive force and by causing membrane leakage. We could maybe get into that sometime down the road, but this basically puts a wrench in nutrient uptake and metabolism of the bacterial cells. And these properties, by the way, are more active in a lower pH environment, so we don't want too much alkalinity in certain circumstances. Otherwise, these effects will be muted. Okay, let's move on to getting bitterness into the beer. And so this is where it gets a bit more complicated. Alpha acids are not very soluble, which of course means that it's not really very convenient for making beer in their native form. Luckily, they can isomerize, and this happens via the assistance of heat into a more soluble form. It takes about 60 to 90 minutes, hence the boil times. And so I've really been trying to decide on how insane to go on this. And without visualization, I think it's going to be really hard to articulate, at least at a very detailed level. So what we want to know is that alpha acids, the way that they are arranged, they're hydrophobic, meaning that they're afraid of water. And of course, this means that they're not very soluble in beer. So it's kind of like oil and vinegar. The oil just moves around in droplets and won't sort of dissolve. And so without some assistance anyways. And so when we isomerize these alpha acids, this changes. So the molecular composition doesn't change, just the arrangement. And I've used the dinner table analogy before about moving guests around for better conversation and less potential violence, and it sort of holds. So basically, the alpha acid has a 6-carbon ring with some side chains. And during isomerization, this ring opens, and then it snaps into a 5-carbon ring. The process exposes a bunch of hydrophilic sites that were kind of locked away otherwise, and this allows the new iso-alpha acids to be more soluble in the beer and therefore impart bitterness into our final product. Just to make it a little bit, I mean, that's the simplest I think I can explain it, but just to make it a little more complicated, now that we simplified it, there's actually 2 isomerized forms, trans and cis. The cis versions are more stable and significantly more bitter. And generally, they come out at a 2.5 to 1 ratio versus the trans. The trans isomers degrade and oxidize more quickly, and so a heavier cis to trans ratio is preferred anyways. However, the trans isomers contribute more significantly to foam stability, which is a good thing, and we will talk about foam stability when we get to talking about foam stability, but for now, just know that hops contribute to it. So I think that that's probably it for bitterness. We'll dive into hop aroma and flavor next time. And until then, you know the drill. Stay thirsty and stay curious.